Okay, thank you everyone for coming. Welcome to our Wednesday afternoon seminar series. Clearly an unusual situation, a little different this time, but I think things are going along just fine. Our speaker today is Dr. Jeff Havig. Jeff is a candidate for our open faculty position in geochemistry and hydrologic science. Jeff completed his PhD at Arizona State University with a dissertation entitled Geochemistry of Hydrothermal Biofilms, Compositions of Biofilms in a Siliceous Sinter Deposit in Hot Spring. After that, he did postdoctoral and visiting scientist positions at the University of Cincinnati and Penn State University. Today, he's going to talk to us about hydrothermal systems, early Earth and life on other planets. What can we learn by studying hot springs? And before Jeff speaks, let me just quickly one thing here. Jeff, it's our tradition. I don't know if everyone can see my screen at the moment. Should be able to. It's our tradition to give all of our speakers a department mug. So consider this a virtual mug, and we owe you one. That's at some time in the future. So let me go back to your screen. So let's do a test before, make sure that your screen is sharing. Right now, I think I see your camera. Jeff, can you share your slides?
Let's see. Hey.
All right, slides are being shared. Audio is working. 26 participants. We're ready to go. Dr. Havig, welcome to the Department of Geosciences at Boise State University.
Awesome. Thank you so much, Jim. I really appreciate it. And thank you all for letting me into your homes, and welcome to my home. I know this is my first virtual remote interview talk, so we'll see how well we can do with this. So I'm going to focus on my research, a lot of different aspects of my research involving work associated with hot springs and focusing specifically on Yellowstone National Park. But I also want to kind of introduce some of my, how I do research and some of the other different places I do my research as well. And so near the end, I'll kind of go through some different places that have nothing to do with hot springs. So, so if you're bored by hot springs, there's more to come, so please stay tuned. First and foremost, I want to thank a few of the amazing colleagues that I have that have helped make my research work. As an environmental geochemist, I thrive with collaborating with other experts. So like microbiologists here in the University of Minnesota, I collaborate with Professor Trinity Hamilton. I also collaborate with Dr. Alicia Rutledge at Northern Arizona University. I have my student Josh Keither to thank. We're going to talk a bit about his research work. I had a student, Sarah Schroeder, who's an undergrad that worked in my lab, who contributed to this work as well. I'm hoping to bring Dr. Alice Bosco Santos over from Brazil to do a postdoc here. So be talking a little bit about her and Tara, now newly minted Tara Jokic. Dr. Jokic is from University of New South Wales, and she was a student of Martin van Kranendonk, who's also University of New South Wales, and we collaborate with them at the Dresser Formation. Professor Kathy Campbell, who's at the University of Auckland in New Zealand. We do a lot of collaborative work connecting Yellowstone to their hot springs in the Tuapu volcanic zone, or the TVZ as they call it. And of course, my good friend Andy Shaya at the University of Cincinnati, and the newly minted Dr. Andrew Gangadine, who will talk a bit about his research that we co-advised Andrew. So a lot of his work focused on Yellowstone work. So, and of course, funding agencies, and definitely thank the Yellowstone Research Permit Office for allowing us the opportunity and chance to do work in such an amazing place. So my goals for this talk are primarily to introduce myself and my research, so you can kind of get a feel for who I am and what I do. I'll give you an idea of my approaches and products, and also to provide examples of my lab's projects and collaborations, so you can get a feel for not only how I think about science, but where I do my science and how that looks in terms of products. So in terms of environmental geochemistry, it's kind of a vague term that can encompass quite a lot. And so I wanted to kind of give you some different descriptions of what environmental geochemistry means to me. And so first off, in talking to to you all as earth scientists, I can get a little more technical. And so what I tell you is there's all these intersections: geology, geochemistry, microbiology, analytical chemistry, and then oftentimes even within these subdisciplines, there's connections like analytical chemistry and geochemistry really combine to make isotope geochemistry. And geochemistry and geology and microbiology combine to make biogeochemistry. And geobiology, which is a really new and exciting field, really combines microbiology and geology. And so for me, environmental geochemistry really encompasses and touches on all of these, includes all of these to some degree. Now, if I were going to explain this to the university president, I would say that what my work basically is at the exciting action zone of where life, water, and rock interact. And that is really the catalyst, because life, turns out, catalyzes most reactions that occur on the Earth's surface. And so that is where I'm really excited with my work and relating it to the rock record. And so to reflect all this wide range of things that we do in my lab, I coined the Modern and Deep Time Environmental Geochemistry and Geobiology Laboratory, or MADEG Lab, just to encompass just give folks an idea of just all the different things that we're really interested in. And in doing our work, the basic flow is we identify a big picture pressing question that we're excited about. And then we identify modern systems that can inform us about some of these questions, maybe provide us answers. Then we go to these sites and collect and integrate a comprehensive data set with contextual samples as well, and then interpret and then publish the story the data have to tell. So it's not about pushing our agendas or our ideas, but to try to find answers for questions and tell the story that the data has. So enough of that. You probably want to hear all about hot springs and the cool stuff that we're doing associated with Yellowstone. So we'll get on with it. So again, we'll touch on early Earth and life on other planets, but focusing on hydrothermal systems here. So quick background, all of you are very familiar with the Snake River Plain and probably how it got there, but for those of you unfamiliar, Yellowstone currently, present day, is right here. And when the plume first formed, North American continent had not moved to where it is, so at the time that it came, this part of North America was over the hotspot, and so it came up under southeastern Oregon to northern Nevada. And then because of the subduction zone up here, the mantle plume head got cut off and sucked behind that subduction zone, and that resulted in the Columbia River basalts. But the neck of the decapitated hotspot, then the North American plate rode over, and so thus we have this bow front of younging calderas moving through southern Idaho through to now northwestern Wyoming, and where we are doing our work with Yellowstone today. And of course, there's Boise, and there's, not to be surprised by the earthquake. You can see there's a lot of tectonic activity that happens around that area anyway. So to get you familiar with how we think about these hot springs, we do a lot of work in a wide range of hydrothermal systems in Yellowstone, including alkaline systems and more acidic systems. And first off, why are there these different types of hot springs? And really the answer is the subsurface processes drive the surface expression. And so to walk you through what happens, you have precipitation occurring in the mountains just next to the caldera. That precipitation then travels through groundwater. It interacts with the magma chamber and the brines and magmatic gases and heat coming from the magma chamber, which creates then the deep hydrothermal source water, which rises. And then as that gets very close to the surface, you can have two things happen. Either nothing, and that water just comes up the surface, maybe interacts with groundwater, and then it's extruded as a hot spring. And that's where you get minimal phase separation hot springs. And typically you have circumneutral to alkaline hot springs, so pH 7 to 9. And the chemistries are all very similar. And you get a lot of silica precipitation associated with these. So think lower geyser basin, upper geyser basin, for those of you familiar. And then, however, if you have subsurface boiling, so the hot water boils before it reaches the surface, you get phase separation. So just like when you have your pasta water boiling on your stove, you have the residual liquid that keeps the pasta, and then you have a steam or vapor phase that's created, and that steam then cools and condenses. So then you have two sources. You have the vapor phase water source and the residual liquid phase water source. And going back to the pasta analogy, if you think of the pasta as like sulfate or chloride anions, that stays in the water. It does not want to be in the vapor phase. And so you get a concentration of certain elements. And that then is reflected in that liquid phase source. But there's also things that want to be a vapor. So in this case, there's hydrogen sulfide coming up from the magmatic gases. And that hydrogen sulfide wants to be gas. So when you get boiling, it strips the hydrogen sulfide, which then travels with the steam, and then you get condensation, you get concentration of hydrogen sulfide in the vapor phase. And that is how you get acidic systems, because that hydrogen sulfide then is oxidized when it interacts with oxygen near the atmosphere, near the surface or in the atmosphere, and you get sulfuric acid. Now, of course, it's complicated in the subsurface. You get things, so you'll get a range of expressions of hot springs that are all predominantly liquid phase, or predominantly vapor phase, or mixing in between. And this gives us our wide range of amazingly different geochemistries in these hot springs. And as an example of how we can use these different geochemistries to interpret what we're looking at, so I have very complicated plots on the left, and I don't want you to have to focus too much. I'll just walk you through what I want you to take home from this. So on all these plots, we have sulfate on the y-axis and chloride on the x-axis, and they're both plotted in logarithmic scale in concentration. And these are all hydrothermal, all hot spring samples are collected in Yellowstone. So the gray samples were all collected by USGS or past work that I've done. And then the color samples or data points are all from more recent work we've done. This is from a publication that's in review right now in astrobiology. And right here, where I'm circling with my pretend laser pointer, there's a lot of data points that pile up right on that spot. So those are all these minimal phase separation hot springs. And so they all have very similar chemistries, very similar chloride concentration, very, very similar sulfate concentration. But as soon as you get subsurface boiling and that phase separation, then you get this wide range of amazingly different geochemistries, going from either you getting the residual chloride increase in chloride concentration from the residual liquid phase, or you get the sulfide concentration becoming sulfuric acid, so you get high sulfate and low chloride where you have vapor phase dominated, and then you have mixing of the two in between. So you have these end members here, and then mixing all in between. So what that gives us is this natural, of course, talking about the sulfuric acid. So the water coming up from deep typically has a pH of 6.5 or greater, so neutral for boiling water is about 6.3 pH. So it's circumneutral to alkaline, up to 9.5 to 10 in Yellowstone. And on sites, green is intermediate pH, and then yellow is the 3.5 or lower, or the acidic. And you see that these acidic sites all have high sulfate concentration relative to the source, and that reflects on increased sulfide concentration turning into sulfuric acid. And the bottom just represents some samples we collected of microbial communities to show that we collected samples that I'll be talking about a bit from both minimum phase separation and phase separation sites. And for pictures, like here's a typical boiling hot spring in the Lower Geyser Basin, and another one here where you see these beautiful photosynthetic mats developing in the outflow channel. Here is a pH about 5 hot spring in the mud volcano area where you have stromatolites growing. So you have a wide range of really cool biofilms and microbial communities to sample. So we can also look at some of the other elements, and based off of our different chemistries, we can kind of predict what's going to happen. And not surprisingly, for example, iron has a high concentration. So again, we have sulfate versus chloride for these plots. And the basic gist is high concentration is large red, down to low concentration is the small black or gray samples. And So for iron, the high concentrations are all in the vapor phase dominated sites where you have very high pH. So you have iron too that's soluble in acidic water. Whereas molybdenum comes to the surface as a oxyanion, so molybdate. And molybdate stays in the residual liquid phase. So you notice that you have relatively high concentrations in the minimum phase separation sites, but then much higher concentrations in that residual liquid phase from the subsurface boiling. And so it just gives you a flavor of how these subsurface processes can really drive dramatically different chemistries. And of course, this has an influence on life. So I wanted to give you a quick description of what I'm talking about when I talk about these different things. So I mentioned biofilm or biocumulus. Biocumulus is a term that I've coined to best describe hot spring microbial communities that we're working on, because most of the usual definitions don't quite encompass everything that involves. So these biofilms are biocumulus. So this is a photosynthetic mat from an alkaline spring in Lower Geyser Basin. These are actually mostly silica. So if you the carbon and the nitrogen in these, as the carbon's maybe 5 to 8% dry weight. So if you dry them out, then you get something that looks like this, which is mostly silica. So they're 80 to 90% dry weight silica, even as they're alive and gooey. So this is really interesting because you have silica precipitating around living organisms and trapping biosignatures, and then that is the pathway to the rock record. So here is an old sinter that's probably about 9,000 years old from a cut face of an old hot spring in the Sentinel Meadows area of Yellowstone. And so here's a photosynthetic mat layer right there. And to give you an idea, so of course I was going to show everyone and let you touch it for yourself, but I also have, so here's a sample so you can see what that looks like, at least a little bit. And so it's light and porous, and as early diagenesis occurs, you get infilling and slowly becomes a rock. So we're interested in what sort of signals might be trapped, if any. And so to get after that question, one thing we've done is do a lot of acid digestions and carbon nitrogen analyses of both biofilms and sinters. And so what you're looking here on the left is concentration of elements, logarithmic scale. And then you have groupings of elements across the bottom. So we have carbon, nitrogen, phosphorus. So I've termed those bio or bioessential elements. So the fundamental building blocks of life. And I have biotrace. So a lot of things should be familiar. Magnesium for photosynthesis, manganese, iron for, all these are involved with enzymatic processes. Molybdenum for nitrogen fixation. So these are all bioessential trace elements that are involved primarily with enzymatic reactions. And then I also selected some non-biologically relevant elements, so elements that are not essential for the function of life. So lithium, aluminum, gallium, arsenic, right? So these aren't essential for life to live, just as a bit of a control to see if these processes all are uniform. So if all elements are coming in at the same, or if there's differences. And so looking a little closer, right? So we have minimum phase separation on the top and phase separation sites on the bottom. All the dots represent individual samples, and the black is the biofilm, and the gray is associated sinter from these areas. And what we noticed is that there's a systematic retention of elements. So notice for carbon, nitrogen, phosphorus, we have 6 to 9% retention of all these for all biofilms. For the biotrace elements, there's a little more variation, but across all these sites, they're kind of falling around the 20% retention. Whereas when you get into the non-bio samples like lithium, aluminum, gallium, arsenic, they're all over the map, right? There's no rhyme or reason to those. And so what this told us is there seems to be a systematic retention of biologically relevant elements, which makes sense because this silica is precipitating around life and potentially trapping biosignatures. And so this gives us a bit of, we can start thinking about these in terms of conceptual model of how do we get a sinter, right? And to walk you through, so we have local rocks, so we have volcanology producing our rhyolites that are local, and we get lots of the elements from those sources. We also have, of course, modern day, we have biota to think about, so bison, trees, meadows, putting in carbon, nitrogen, phosphorus as sources. those all contribute to making local soil, which can blow in every day when solar radiation drives winds. We also have the hydrothermal water coming from deep, which also contributes elements. So those then all contribute to making up a biofilm or biocumulus. So like photosynthetic mats, chemotrophic filaments, but these over time gain more and more silica precipitates, the streams will deviate, the outflow channels, and so you'll get abandonment of a channel, and then these dry out. So as they dry out and then get buried by new layers of biofilms growing, you then get these layering that eventually comes through early diagenesis, these centers. And if you zoom in on these centers, you can see the microbial mat textures preserved. And so if you look inside of the lower right, here's the scale bar at 10 microns, those little tubes, those are all where biological filaments were. So cyanobacteria, to be specific. The cyanobacterial filaments were in those spaces. And so we have preservation of those spaces where the filaments were, we have preservation of the sheaths that formed around those, and we have starting to have infilling of the spaces through early diagenesis. So this is great for like a very broad spectrum, wide view, right? Even the widest of views where you're just taking a rock and digesting a big slop of biofilm and digesting it, we seem to see evidence for biosignature. So we wanted to zoom in, and this is work that Dr. Gangadine worked on for his PhD dissertation. And so here's an SEM image of a siliceous center. And again, I want you to see those little, so here's the sheath, right? These are all the sheaths, the silica sheath, and that space inside the sheath is where the filament was. And so we have little colloidal silica precipitating on the outside. And here we can see this part has gone over minimal diagenesis so far and minimum infilling. And so what Andrew did was he used the biosims, which is a dual beam secondary ion mass spectrometer that has the cesium and oxygen beam. And what that does is it allows you to analyze pretty much just about the whole periodic table on thin sections at the same time. So you can switch between beams. And so you can look for carbon at the same time you're looking for trace elements, for example. And that's exactly what Andrew did. And so, and lo and behold, not surprisingly, we found where there was carbon. We also found, so this space represents where a filament was. So this would be the empty hole inside the sheath. We find carbon, we find nitrogen, we find sulfur, we find iron, you can find aluminum, interestingly. But something else we noticed was gallium, which life shouldn't give a rip about. Gallium seemed to form these halos around the sheaths. So here I'm pointing out with the virtual laser pointer, here's where the filament was. This is that sheath. So the silica sheath that formed around the filament. And in cross section, here's that ghost of the original sheath. And so maybe this might be helping inform us about the processes where these sheaths are first precipitated, and we can start to think about, well, what happens through diagenesis? How do these change as you get heat and temperature altering these? And Andrew's following up this work with some hot spring deposits from 400 million years ago, and hopefully we'll also be able to apply this to some older hot spring deposits as well. So we're tentatively optimistic that maybe we're getting somewhere with this idea. And of course, we're applying these to sites in New Zealand. New Zealand is really interesting because whereas the Yellowstone magma source is relatively low at the TVZ, it's actually very close to the surface. And so what we think is happening is that's driving that subsurface processing to a much greater degree. And so this gives us much greater extremes in geochemistry at these sites. And so we're excited to see, can we do these initial things we're finding in Yellowstone also apply to other sites? And of course, you have to have it. So there's lots of great places we're going, lots of tea trees to wade through. Of course, it's a little different. We can just do work as long as we get permission from the Maori that own the sites. If they give us permission, we're allowed to just work right in the view of public, that's a no-no for Yellowstone. And of course, I have to have a picture of famous Champagne Pool. If you ever go, you should definitely check that out. It's just gorgeous. And I also want to include this picture in the lower right. So this is the Waimangu geyser. So in Maori, Wai means water. And so you notice Wai o tapu, that means sacred water. Here, Waimangu means black water. You notice the black stuff blowing out. This is actually a geyser, Waimangu geyser. and it was erupting until 1904. You notice these two characters right here. Here's our Earth scientists, of course, running towards the massive hydrothermal eruption going on. So, you know, Earth science, we're always running towards danger to find cool things, right? Now, another really exciting bit of research that we're doing is with, what I had mentioned with Dr. Jokic and Professor Martin van Kranendonk, is looking at the Dresser Formation. So Tara's dissertation work focused on a reinterpretation of a site as a terrestrial hot spring. So it's hot spring deposits that formed at 3.5 billion years. This would blow out of the water. The oldest, you know, currently the oldest sites are about 400 million years old. So this is amazing opportunity to look at really early life in hot springs. And she has interpreted, so this is the Dresser Formation. That black line along the top of the ridge that I'm showing is a chert layer. And that would have been the paleo surface at the time of this being a small, like 20 kilometer-ish diameter caldera, similar to Yellowstone, but just smaller. And if you're from Arizona, the superstitions are a similar size caldera. But so this chert was forming as hydrothermal deposit. It's been tilted away from us as you're viewing that picture. So the top is tilted down and away. And what that has allowed is actually exposure of the subsurface plumbing, which is, when I got to go there 2 summers ago, it was just like amazing to see this, because it was like looking underneath Yellowstone. So if you look at all these red dikes coming off, those are all feeders for the hydrothermal system. And as you look closer and closer, they're all crisscrossing and talking to each other. So it's just really cool to see our ideas of how the geochemistry works in the subsurface laying out to bear. And it was happening 3.5 billion years ago. So just awesome. And while we were there, I found this character. So this is a stromatolite that was forming at the edge of a hot spring. Here's my finger for scale. And it looks just like stromatolites that I'm finding in modern day Yellowstone. So it's very cool. And of course, they let just any hobo ride in back of the utes and across the outback. So that was a lot of fun as well. But the challenge with Tara's interpretation is proving that it's a terrestrial hot spring. So this is just one bit of evidence that we were able to glean using Yellowstone as a modern day example. So here I am standing on the rim of an extinct hot spring. So this rim is the edge of the hot spring from when it was active. So this would have been inside where I'm pointing here. And what we found is, so Tara had this sample that she had collected from her site where you have all these bits of sinter that all kind of piled together. And what I found this site here by this extinct hot spring with exactly the same thing, with these pieces of sinter with all this infill of silica sand and deposited amorphous silica. And so as we zoomed in on these, we found that they're identical. You have this rim of thicker silica, and then inside this, it's more porous. If you zoom in on the boundaries have identical structures, and if you zoom in on the interior, so here's amorphous silica that's, you know, tens to maybe hundreds of years old, and here is what was amorphous silica 3.5 billion years ago, or at least that's my feeling. And the textures are the same, it's just been recrystallized. And having this kind of preservation, again, is mind-boggling. That's coming out here in astrobiology issue that Tara submitted as part of her dissertation work. Some more really tantalizing evidence. So here is some of this geyserite, or putative geyserite. So to form geyserite, you have to have hot water precipitating at the water-air interface. You can't form geyserite in the below water. So here's from the site. So if you zoom in on the geyserite sample that she collected from here, here's a zoom in picture, and I have same scale, then a hot spring from Yellowstone. This is Rosette Geyser, one of my favorites. And so along the edge of the hot spring, you see this white stuff, that's all silica. And that's all silica precipitating from this boiling hot spring. And if you take a piece of that silica, break it off, and make a thin section, here's a thin section, same scale. And here you see, hopefully you can see these very similar textures in both of these. So to me, this is also very, very convincing evidence for me, and hopefully for you as well. And so one of my evil plans is to submit with Tara not this spring, but the next spring, submit a NASA exobiology proposal to fund her, bring her to the US, and and really push hard on some of this preliminary work she's done, and get her in Yellowstone and really make some connections with this and push hard on it. So hopefully we can shake some loot loose to make that happen. Also with the dresser, we're in our collaboration with Martin Van Kranendonk here and Kathy Campbell and their student Michaela Dobson. They're working, and Clark Johnson from University of Wisconsin, and Mike Rowe, who's also University of Auckland, and a bunch of other really amazing students and colleagues. So at the Dresser Formation, they drilled into the formation. So the idea was that if we have lots of samples from weathered outcrop, but we want fresh samples. And this is a huge gamble because, of course, hot springs are very localized, right? So if you're going to drill for a hot spring, you know, if you're off by a foot, you don't get a hot spring deposit. And fortunately, they hit the jackpot and nailed a hot spring. And so here on the left side, I'm holding a piece of core that has some, what I think is geyserite preserved in it. And here it's a pretty green instead of that oxidized red. And to the right, these little buttons, these little knobs here are actually small stromatolites that are preserved with pyrite, something we wouldn't see on the surface because it would have been oxidized away. And so we're really excited about some of the work that's going to come out of this from Michaela's PhD project. So stay tuned with that. That's some more really cool and exciting work coming out of this. So work that my current PhD students is doing is focusing on hypoliths. So hypoliths are exactly what it sounds like, below rock. So if you have a layer of rock, you'll find oftentimes life living underneath some gravel or a piece of quartz. So if you're ever in the desert and you lift up a piece of quartz that's on the surface and look underneath, you'll find that it's green. And that's because you have hypoliths or photosynthetic communities living under those, and they're protected from desiccation, they're protected from UV radiation by the quartz. And it creates a little microenvironment for the live. There's enough light making it through the quartz as a window that they can still drive photosynthesis, but they're protected from desiccation and UV radiation. So when you're looking at a hot spring, you go to, so this is what we call smoking gun hot spring. And so here's the source and the outflow and all this beautiful set of photosynthetic mats. If you go to here to do research, of course, you want to run up to the hot spring and sample that, because that's the cool, exciting thing. But what I want you to look in this image, which is Google Earth image. So this is a 40-meter scale bar. Look at all the white. So all that white is just silica. So it's all amorphous silica that's been precipitated by this hot spring over probably hundreds, maybe even thousands of years. And it's deposited silica as this stream is meandered back and forth. And this gets broken up from freeze-thaw cycles, from bison walking across it. And so you get this gravel, sandy gravel. And you'll find this in all hydrothermal systems. But something I noticed when I was working on my dissertation is that at the end of the day, oftentimes you have some extra deionized water, and rather than carrying it back to camp, you just pour it out. And what I noticed is when I pour that out, it kicked up green, which seemed very strange. And so I noticed when you look closer, you start scraping away some of that white center, you actually find green underneath. And this is, I found through work, now more and more work, that it's everywhere. So acidic sites, alkaline sites, it's everywhere. And so we were interested in this and to characterize this because this had never even been discussed in literature at any time. And everyone just walking over and ignoring it. So we thought, maybe this is actually informing us of some ancient times. So during the Archean, we have a lot of geochemical evidence for the presence of oxygen before the Great Oxidation event. And we have evidence going back to about 3 billion years ago of surface weathering involving oxygen, even though there's no evidence for oxygen in the atmosphere. So we have this 500 million year mystery of how can you oxidize a continental surface without having oxygen in the atmosphere. So maybe this is one of the solutions, right? So if you have some silica sand, if you have hot spring deposits making amorphous silica, you can have wide swaths. I mean, this is a huge surface area for one little hot spring, huge surface area where you can have a photosynthetic mat. And that photosynthetic mat can be generating oxygen. The oxygen can either seep in the subsurface below the mat, or it can get washed into nearby streams. And then just a whiff, the tiniest whiff of oxygen will kick off the party for oxidizing pyrite. So you can oxidize pyrite, generate sulfate, start dissolving carbonate rocks, and really kick off the party. And basically what you have is perfect conditions, because you have protection from desiccation. It actually protects from being washed away by water as well. The gravel protects from rain, and also protection from UV. So even that small layer of amorphous silica attenuates UV radiation, so it protects them. So, which is a problem during the Archean. We think that the Earth's surface was bathed in UV radiation. So here's another solution for that too. And so Josh, we'll be submitting a NSF grant to Low Temp Geochemistry and Geobiology to help fund Josh's continued work on this project. So one final thing for looking at hot springs, another fun story, is thinking about, well, early Earth metabolisms. And that's a real interesting question that we grapple with, is how did life very early on get its energy? How could it drive metabolisms, right? You have to have redox gradients for life to take advantage of, to move electrons, and then to take advantage of moving those electrons to fix carbon, for example, or to make molecules and generate ATP for what we do now. So it's a really interesting conundrum. And I think terrestrial hot springs provide an answer that is right in front of us. So this is Dante's Inferno in the Sylvan Spring area. And so this is gas. This is not boiling. This is actually at about 78 degrees Celsius. So it's not at boiling. That's all volcanic gases coming up. So this thing's really cranking with volcanic gases. And we think it has a molten sulfur layer down deep. And so these gases are kicking up molten sulfur. So if you look at these SEM images to the lower right, so zooming in and looking at C and D, those globules are actually elemental sulfur globules. So sulfur is hydrophobic, so it makes a sphere to minimize its surface area exposure to water. But what we notice when we zoomed in, if you look really close at these here, so all these scale bars are 10 microns. These globules are getting, look how lumpy they are when they get smaller. They're getting chewed up, they're getting oxidized. And you can see this, hopefully you can see this little halo of gray. So desiccation has kind of wrecked the biofilm, but these were coated in biofilms. And you can see these little channels around them. That's where cells were attached to the sulfur globules and oxidizing them. So we have evidence for sulfur oxidation in the spring. What's really cool, looking over to B of this, so the upper right of this figure, and then going down to E and F, we're zooming in those white things in this SEM image, those are pyrites. And you notice that there's this darker gray biofilm that's tightly associated with those pyrites. And so what we have here is a different microbial community in the same sediments, in the same hot spring, that are reducing elemental sulfur to sulfide and precipitating as iron sulfide. And we have evidence that they're also probably reducing iron 3 to iron 2 as well. So here we have two different microbial communities doing two different reactions in the same hot spring. So this gives us some really exciting look into how we might have some cryptic sulfur cycling occurring in these hot springs. And maybe this is evidence and hints as to what life was doing very early on. And this has some really interesting implications for thinking about other planets as well, because if early Earth had these hot springs giving this free energy for life to use, if we're thinking about looking for life on, say, I don't know, maybe Mars. Curiosity is currently exploring Gale Crater. So here was the landing ellipse for the Curiosity rover, and it's heading towards the peak. And here's Curiosity rover here. A colleague of mine, Amy McAdam, who we did grad school together at ASU, she and her team analyzed a lot of elements using the mass cam, but they are also able to analyze specifically for the redox state of certain elements. And so in this image, which hopefully you can see, I'll walk you through. So in this image, we have intensity on the left, and then we have wavelength on the x-axis. So we have orange. That orange, it represents reduced sulfur or sulfide. And we have this red peak. So here you see orange and then red at the same sample, orange and red at the same sample. That red represents sulfur dioxide, which would be the indicator of sulfate. So here at the same exact place in these veins in this lake deposit that's about 3.8 billion years old-ish, we have evidence for reduced and oxidized sulfur in the same place. So for me, that's a tantalizing hint that Mars had the right conditions to really support life early on, and especially in these hydrothermal settings. And another one of my evil plans is to submit a proposal with Dr. Alice Bosco Santos to bring her from Brazil to work, to drive on this and to start looking to tease apart, like, are there are there sulfur isotope signals that are imparted by these processes? Can we start to tease apart biological signals from these processes in these hot springs? So hopefully we can shake some loot loose this spring to bring Alice to the US and start really pushing on this project. So that wraps up the hot spring portion. And so now I'll go on to just to touch on some different types of projects that we're working on. And hopefully you'll notice that although they're wildly different from these hot springs and earlier things I was just talking about, they all kind of encompass the same idea of looking where life and geochemical processes, where they interact with each other and can produce signals that can be locked in the rock record, or where they come together to react and drive processes that might be impacting us today. So, excuse me, I'll start off with Merrimack Lakes, and specifically Fayetteville Green Lake, New York. So here's Green Lake. It's just outside of Syracuse. So those of you familiar with upstate New York, excuse me. This, of course, Green Lake's beautiful. It's a place where a lot of Syracuseans go to enjoy the beach, where an artificial beach was put in. But the really cool thing about this place is its geochemistry in the subsurface. So we collaborate a lot with Mike McCormick. So Professor McCormick's from Hamilton College, which is a liberal arts college in Clinton, New York. And they have this pontoon boat that they're allowed to take out on Green Lake. They're the only ones who can do this, where they do a lot of work with undergrad research. Unfortunately, they've been allowed us to join them and work with them. And Mike has developed this amazing system for collecting instantaneous entire water column samples. And so what this is effectively a fancy garden hose, where you cut a hole in the hose, and you take a 60 ml syringe, and you attach a tube to the end of the syringe, and at the end of that tube, you put a glass pipette, and they carefully heat up and bend the tip of the pipette, seal it off, and then they're able to draw a vacuum on the 60 ml syringe, put in a pin to hold that vacuum, tie that to the garden hose, fix the pipette tip through the hole, right? And then they do this about 70 times as they lower this hose through the entire 53-meter depth of Green Lake. And then you send a messenger down the garden hose, it breaks the tips, and boom, you have water samples through the entire water column all at the same time. And then we quickly pull that up, take off the 60 ml syringes, put them in coolers, run off to the lab, and quickly process all of those, filter them, and process the samples for analyses. And so this allows an unprecedented look throughout a water column of what's going on with the geochemistry. And we use this to great advantage, I think. So a little background on Green Lake, it's meromictic, which means it does not turn over. So because of the water coming in, so it's fed by groundwater that's saturated with respect to gypsum. And so there you have high calcium, high sulfate concentrations, and it generates this layer where you have oxic on top in the top 18-ish meters. Then you have a chemocline where oxygen goes away, and then sulfide comes in. And in that chemocline, there's a turbidity peak, and that turbidity peak is purple sulfur bacteria. So that purple sulfur bacteria is called a plate, just live right at that boundary between these two chemistries. And you can see that the sulfide concentration gets close to 1 millimole concentration, very high. So there's a lot of sulfate reduction going on. And what this does is it provides us a really cool proxy for the Paleoproterozoic ocean. So we think that around the time of the Great Oxidation Event, all the pyrites on the continental surfaces were all rapidly oxidized. You had a flush in of sulfate. That sulfate was being reduced by sulfate-reducing bacteria. And we think then we had oxic water on the top of the Paleoproterozoic ocean and sulfide at the bottom. So here is a little window to give us some idea of what was going on with the chemistry there. So we want to tie this in with another interesting question. And that question is, how do you get these manganese carbonate ores? So most manganese carbonates or manganese ores are manganese carbonate. So rhodochrosite, manganese carbonate, or kutnohorite, which is calcium magnesium carbonate, and really pretty pink minerals. But how do you get these, right? How do you form this? And we think that we might actually be able to provide an answer. And the reason why this ties into Paleoproterozoic ocean proxy is because the world's largest by far manganese carbonate ore body was deposited all at the same time right after the Great Oxidation event. So we think we may have had an instance where we had the kind of ocean like Green Lake. And if we look at Green Lake chemistry and zoom in, what we see is as you go transition, so here's from 15 meters to 25 meters in the water column. So as you transition from oxygen going away to sulfide coming in, we have this peak in manganese concentration right at the microbial plate. So what we think is happening is you have manganese oxides, which are insoluble, settling down. They run into this zone and they become reduced because manganese 4 is a great source to dump electrons onto. So it gets reduced to manganese 2 and diffuses up, gets oxidized, cycle, and this cycling then generates this big peak of manganese. And coincidentally, we have supersaturation of manganese carbonates right at that peak. So what we think is happening is we have cyanobacteria at the surface driving whitings. So when cyanobacteria convert bicarbonate into carbonate, or CO2 into bicarbonate into carbonate, and take the CO2, they kick off hydroxides, and so they raise the pH. And so the pH gets raised, you have lots of calcium, lots of carbonate, you precipitate calcium carbonate, those calcium carbonate crystals then settle down and then go through this layer of high concentration manganese and build a rim of manganese carbonates. And so to test this, we collected sediment samples and took them to the synchrotron, and Beth Herndon drove this research. Professor Herndon's now at Oak Ridge Laboratory in Tennessee. And it's always great when a hypothesis actually pans out. So what you're looking at the bottom right is this blue, that's calcium. So there's your calcite crystal, original calcite crystal. And then around the rim, you see this turquoise. This turquoise color is a combination manganese and calcium. So essentially kutnohorite. And we see from looking at these crystals that they line up very well with rhodochrosite standard. And so we have evidence, proof that this is the kind of process that's happening in the Green Lake water column. And so EVIL plan is to submit a NSF proposal to low temp geochemistry and geobiology to see if we can drive this further, to see, well, can we get ore body concentrations, right? Right now, the concentrations are not ore concentrations of Green Lake. But if we ramp things up and think about, well, what about scales of an ocean? Can we get the kind of values that would produce an ore body like what happened after the Great Oxidation event? Also interested in harmful cyanobacteria blooms. This is something that's been happening across the country, across the world. Reservoirs, lakes. On the upper left, we have an image from 2011 of a cyanobacterial bloom. The green here that I'm highlighting is all cyanobacterial bloom in Lake Erie. So you can see them from space. They can be so big. And what you have is cyanobacteria blooming due to high concentrations of fixed nitrogen and phosphorus being delivered primarily from agriculture. And so what they do is they drive these cyanobacterial blooms to generate a lot of biomass, and that's their strategy to choke out and take all the elements to outcompete other organisms. So they create all this biomass, and that biomass gets then settled in and sequestered into sediments. But we were thinking, well, what about the impact of invasive mussel species? So quagga mussels and zebra mussels have all invaded Lake Erie and a lot of other reservoirs. And so we looked at some that have mussels and don't, because it's known that quagga mussels will filter out a lot of the cyanobacteria. And so this is the conceptual model that touches on what we found. And the basic gist is when you have cyanobacterial blooms in the top, what you have is a lot of organic carbon being deposited. But with the introduction of invasive mussel species, things switch a bit. So you have less organic carbon being sequestered in the sediments, You have a more complicated carbon system going on, but you get an increase in carbonate from the shells. So it's definitely changing. And why does this matter? Well, it turns out that lakes and ponds, and even that Farmer Fran's pond out back, lakes, ponds, they actually sequester more organic carbon than the world's ocean by about 3 to 4 times. So they're actually hotspots for carbon sequestration. And we're changing these hotspots, and we don't really understand how we're changing them by introduction of invasive species and driving cyanobacterial blooms. So it's more work that we need to do to better understand what's happening to our system as things dramatically change. Finally, I want to talk about some of the work I'm doing on glaciers, again, something completely different. And again, thinking about carbon cycling and trying to better inform our ideas of what's happening with the carbon cycle and what's happening with global climate change. And we were interested in looking at these to understand how these microbial community systems are impacting glacial surfaces. Lots of recent work has shown that as you get these blooms, so you can get algae or you can get cyanobacteria. For the systems we're looking at, they're primarily algae. You notice, hopefully you can see this red splotch. That's not a crime scene. That's actually pink snow. So that's an algal bloom that's occurring on the surface. And what happens is, as it turns pink, of course, pink is much darker than white. And so actually lowers the albedo at surface and drives melt. And there's a study done in Alaska that actually connected snow algae blooms driving up to 11% of all melt of snow and glacial ice during one melt season, just from algal blooms. So this is a huge component driving the loss of glaciers, and these alpine glaciers especially, but this is also happening in Greenland, and it's starting to happen more and more in Antarctica, so this is a real thing that we need to be more aware of and understand better. And of course, this has some really interesting implications for deep time as well, right? Because of course, we're in a glacial period, and there's also the Neoproterozoic, right? So here's the Kingston Peak Formation near Pahrump, Nevada. Diamictite, I'm pointing at a big old rock right there, cobble. And then going deeper in time, right after the Great Oxidation Event, about 2.45 billion years ago, the Earth was rocked with 3 to 4 global glaciation events. And here, Dr. Jokic is pointing to a drop stone in a deposit that it represents the very first one of those ice ages immediately after the Great Oxidation Event in Western Australia. So we can start thinking about these snowball events, and maybe we should be thinking about them a little differently. So here's some snow algae I'm collecting with a spatula. You know, the pink hue, how much darker that is in the surface. Maybe this actually has implications for snowball Earth events, right? If we're thinking of a planet covered in snow and ice, this is actually an ideal habitat for snow algae. And maybe as you increase CO2, you drive more algal blooms, you actually could darken the surface. And maybe this is a positive feedback loop that plays into ending of snowball Earth events. And we're also interested in trying to understand these systems before they go away, right? And as they influence the loss of alpine glaciers. Because where I come from White Salmon, Washington, which is right about here, right between Mount Adams and Mount Hood, right in the Columbia River, where I grew up, and all across the Pacific Northwest and many other places. Communities depend greatly on glacial melt for water sources during the summer and for groundwater recharge. And so as these glaciers go away, they're melting very rapidly. Some back of the envelope calculations I made is a lot of, like, Mount Rainier may not have glaciers in 80 years, depending on how rapidly they retreat. So this is a very real problem that's happening very fast. So we need to understand what's driving them, what's happening, what's even on these communities. We're losing biodiversity that we don't even know we have. So what are we even losing from these systems? And so it's really pressing to think about these. And another really fun aspect to think about is, again, looking for life on other planets. So it turns out our neighbor, Mars, is a glacial planet, and has been a glacial planet for about 3.5 billion years. And here's a glacier image, a high-rise glacier image from NASA, looking at a glacier coming down. And here's the lateral 3D rendering of that glacier. So here's a glacier coming down, here's end moraines, lateral moraines. So maybe we could even think about these snow-adapted systems as a type of life to look for when we're looking for life on other planets. So to wrap up, and of course, there's lots of other fun things that I'm working on that I wasn't able to talk about just due to time constraints. Like for example, some really cool acid mine drainage work that we did in Kentucky and connected to the Great Oxidation event and how that may have been driven. Looking at the Medicine Bow Mountains in Wyoming, and there's some amazing stromatolites. That's just some geology candy there. And look, that's my rock hammer, so evidence that I do actually go out and use that rock hammer. I'm not just a chemist pretending completely, mostly pretending, but not completely. But we have some really cool carbonate rocks that we're exploring there that show the end of the greatest carbon positive isotope excursion in Earth history. And so, that tying with all the other things I'm working on, hopefully you've got a good feel for the sorts of things I'm interested in, the kind of things I'm interested in exploring. And of course, I have not talked about a lot of backyard projects. I'm really excited about taking these tools. They can be applied to anything and anywhere. So I'm really excited to apply these tools to problems specifically for Idaho as well. And I think hopefully you can see how this toolkit can be applied to just about anywhere and ask some really interesting questions and hopefully answer some really interesting questions. So thank you so much for your time. I'll leave you with a picture of Old Faithful erupting. I took this, or a movie of Old Faithful erupting right after a thunderstorm went by. And thanks for inviting me into your homes and for giving me a chance to share my research with you.
All right, 1 second here. I don't know if this is coming across. That was supposed to be an applause.
Yeah, I appreciate it. Not a lot of applause around here.
Well, if you were
I'll just
here,
pretend everyone hasn't fallen asleep.
Yeah, thank you very much, Jeff, for the talk. We do have some time for some questions. I think the best way to do the questions is if you have a question, raise your hand in your chat in your screen. To do that, you'll see a participants button. If you click on that, there'll be a raise your hand option. And then as you raise your hands, I'll call on you. I think you'll sort to the top of my screen if your hand is raised. And so we'll go that way. And I guess as long as I'm talking, I'll start with a question.
Sure, absolutely.
I'm curious about let's see, in the Green Lakes, that turbidity layer. I didn't quite follow. You broke up just a little bit during that.
I'm sorry.
So no, it's my internet connection, I believe. Generally, it was great, good connection all the way through. So the turbidity layer, I didn't follow that. And a couple of slides later, you were talking about that microbial plate zone.
Oh, yeah, that turbidity layer that we measure with our sonde is just a high density of purple sulfur bacterial cells.
Okay, I mean,
and actually, if you send a camera down through the water will be pretty turquoise, a little darker, and then it'll just turn into this red cloud layer as you pass through the purple sulfur. It's really cool. And it's really constrained right to a very tight boundary between the oxic and the euxinic water.
So, I mean, essentially suspended particulates then? I mean, what is keeping it at that layer? Is it just a temperature-related density differences?
There are temperature and density related differences between those layers, but they are actively staying at that because purple sulfur bacteria actually they don't do photosynthesis like plants do, or cyanobacteria. What they do is anoxygenic photosynthesis. And so they use hydrogen sulfide instead of water. So they oxidize hydrogen sulfide to sulfur. And so they're right at that boundary where there's enough sulfide coming up that they can take advantage of that and be at that sweet spot, but high enough to still get enough photosynthetically active radiation. And so these sorts of plates are very common at these meromictic lakes.
Interesting. Okay.
Yeah, they're very cool.
Thank you. So looks like Mark was the first one with his hand up. I'll unmute you, or you can unmute yourself, Mark, and ask a question.
Okay, great. Thank you, Jeff.
Absolutely.
Curious, what is ultimately limiting the snow algae or cyanobacteria in terms of their productivity and volume and importance for albedo?
Oh, wonderful question, and a fun yet tragic story. So one thing we were interested in is exactly that. It's trying to tease apart, are they limited by nitrogen? Are they limited by phosphorus? You know, what's driving the blooms? And we went to the Pacific Northwest specifically because there's no carbonate rocks nearby. And so the only carbon they're getting is from the atmosphere. And so it really helps to limit limit where the carbon's coming from. We can trace a little better. So we did carbon uptake experiments. And with the carbon uptake experiments, we add a 13C labeled carbon, so bicarbonate, but we also add then fixed nitrogen or nitrate or ammonia or iron or phosphorus. And what we found is everything was stimulated, which doesn't make any sense. Like, well, one thing should come out better than others. And then it dawned on us, oh yeah, we're adding carbon to all of the samples. And so we went back the next season and did an experiment where we just added increasing concentrations of inorganic carbon as that bicarbonate, and we got a linear increase in photosynthetic rates and carbon uptake as we increase concentration. So the cool thing is we found out, oh, they're CO2 limited. The tragic thing is they're CO2 limited. And of course, we're pumping CO2 in the atmosphere, so perhaps this is a positive feedback loop where more CO2 in the atmosphere drives more of these blooms, which drives more melting, which drives more rapid loss of snow and glaciers. So yeah, thanks for that question. It was a cool yet sad story.
Yeah, no, thanks. That is really interesting.
Okay, thanks, Mark. Jen has her hand up.
I am going to.
Oh.
Jen, are you? I might have muted you. Hang on.
All right, can folks hear me?
Yep, we can now.
Uh-oh, now I can't hear any of you.
You're good.
I'm going to turn this
I can hear you.
I guess my great talk and very diverse interests. So thank you very much for that. My question is about a couple things. I'd like to hear more about your acid mine drainage work and specifically whether the speciation of iron in those can be related to natural or anthropogenic acid mine drainage. And also just wanted to confirm, you said that there was a lot more organic carbon in lakes than in the oceans, which is amazing.
Yeah, and that's something I learned as I was getting more and more into lakes and we started doing the project really a lot of groundbreaking, wonderful work by folks in Wisconsin and actually in Minnesota and some other places where they've been doing a really great job of trying to nail down just how much carbon is being locked into sediments in lakes. And yeah, as far as we can tell, lakes are really big actors in terms of depositing carbon, especially organic carbon. But at the same time, it's a time scale difference as well, right? Because a lake is a relatively transient thing. So it's really important for a short term, but the ocean has that hundreds of millions of years of locking away carbon advantage. So it's yeah, really fascinating learning about that. But so the acid mine drainage, so you're interested in the iron?
Yeah, I was interested in both the speciation of the iron and the sulfur. To my understanding, some of that has actually been used in lawsuits to show that some of these mine sites had always produced acid mine drainage and therefore couldn't be attributed to mining practices.
Right. I'm not familiar with the legal ramifications, so I definitely don't want to step into that can of worms. But we weren't really using the best tools for teasing apart speciation. We have a pretty good idea what was iron 2 and what was total iron. We were more interested in tying iron oxidation with the microbial communities, and maybe the microbial communities might be something that plays a part as well. One interesting thing that came out of that was looking at what we found is we had a wide range of pH values at a lot of different sites, so pH 2 to pH 7, but we had one organism that was dominant in all these systems. And it was a thing that could do carbon fixation, do nitrogen fixation. It could oxidize pyrite. It could oxidize iron 2, and so basically and it could live at pH 2 to pH 7. So it's this amazing microbe, which has really got us thinking about thinking of the great oxidation event, because here's an organism that you just give it a whiff of oxygen and it is off to the races, and it doesn't need anything else. It just needs a whiff of oxygen and some pyrite, and it can crank. And so that was one really cool thing that came out of that, which with some follow-up work. In terms of, I think maybe sulfur isotopes might help as well, which we did not do any of the isotope work, like iron isotopes or sulfur isotopes. So I think that would be really interesting to look at those sites. Those were, what we were looking at was historic coal mining sites in south central Kentucky in the Daniel Boone National Forest. So abandoned mines for coal. That's a really interesting question. I want to learn more about that now.
Thanks so much, and thanks for a really amazing talk, and look forward to talking with you more tomorrow.
Yes,
so thank you.
Yeah, absolutely. Thank you.
Okay, I don't see any other hands up. Is there anyone trying to ask a question and can't figure out the hand? If so, go ahead. Oh, let's see. Jenna.
Yeah, it was really interesting talk. Thanks very much, Jeff.
Thank you.
I'm curious. I look at well, historic tertiary hot spring deposits and centers, and I really liked your microbial textures and elemental distribution. That was interesting. I'm curious if some of that biosims and other work is able to distinguish probable different types of microbes in some of these different chemically different systems.
Boy,
and also about what's the rate of infilling to go from the textures in the current hot springs to a 9,000-year-old center to a 9 million-year-old center. Any ideas?
For the second question, I think that's one of the $60 million questions that I would really like to try to figure out. But there's lots of course, remobilization of amorphous silicates dissolves and reprecipitates. And so there's some transfer from upper layers to lower layers as it's infilled. So I think that's a really interesting question to try to think about moving forward and thinking about early diagenesis. But for the previous part, that is really one of the most exciting things that I really hope to learn more about, is trying to see not only can we say there was life, but actually inform ourselves about what kind of life was present. And that's why we're looking at, for the very first order look of looking at minimum phase versus phase separation sites, to say, well, are there differences? And there were some subtle differences between those, but that's of course, that kind of is a bulk community. Now, if we can look at the microscale and start to tease apart, well, here's something that's specific for photosynthesis, or here's something specific for chemotrophy, or here's something specific for for a certain metabolism, a sulfur metabolism. Boy, that would be amazing, because then that's something we could use to really start to learn more about what we're looking at in the rock record. So, that's one of the really exciting things that I think I'm hesitantly optimistic there's hope to get to that point. So yeah, thanks for that question. It's exciting stuff for me.
Great. Okay, let's go to Leho.
Hey, Jeff. Thank you for that. Yeah, I enjoyed your talk. One of the things that I sort of frequently think about is, and a lot of people think about is representing sort of biogeochemical processes in land models, and in particular, sort of how we do a better job at representing, in particular, the coupled cycling of carbon, nitrogen, and phosphorus, and how those are sort of tied to or constrained by, things including like, molybdenum. And microbes have come up, significantly in the past sort of few years about sort of these so-called microbially enabled biogeochemical models in land models like the community land model. I wonder, from the, your research is, yeah, I guess I'm wondering sort of how what opinions you have on sort of that as an emerging field, and how some of the work that you do sort of might help kind of inform some of those efforts to sort of advance the fidelity with which we represent biogeochemical processes in land models.
Yeah, I'm really excited about, there's so many new modeling techniques that are starting to really, I think, do a really great job of incorporating microbial communities and microbial metabolisms into them now. Now that we realize more and more the microbes are everywhere, and they got their fingers in just about every chemical reaction. And I kind of see my job as someone that can quantify what's happening, so we can quantify carbon uptake rates, we can quantify nitrogen fixation rates, we can quantify how much of a microbial community is doing certain metabolisms, and try to inform, provide data that then is useful for modelers to then fine and tune in, just like you say. So I kind of feel like I'm a feeder for modelers to give them better and better information to run with, hopefully.
Great, thank you.
Hope that answers the question.
Yeah, thanks.
Okay, any other, I don't see any other hands up, but if
Oh, yeah.
oh, another one. Okay, go
I'm-
ahead. That's
I'm
Mark.
back up.
Okay, I thought you just didn't put your hand down.
No, I figured that out, that I could put it down, but I put it back up. And it seems like this is a great forum to ask questions forever.
Yeah,
We're all just sitting around at home anyway. So.
everyone's home, hopefully in a comfortable chair.
I just quickly, you know, Bill Shop's claims of the microfossil evidence of life in the apex chert have been controversial for decades. It seems like the rocks that you're looking at, I guess I'm curious as a question, what are the relationships between the formations that you're looking at compared to those locales? And I mean, do you agree that it's hard to not argue that there is direct evidence for these prokaryotes at that time?
A lot of course, for those of you not familiar, that's a very hot topic for microfossil community of shock versus everyone else, it seems. With a lot of his early work, I was pretty skeptical because having been out to the apex chert and seeing where samples were collected, I think that the samples he collected were transported there for somewhere else and definitely processed quite a bit hydrothermally. So I'm a little skeptical of those, but really those arguments that have gone back and forth between like Brazier and Schopf and all this infighting is kind of what's inspired me to try to think of it, try to come up with a new way to look for evidence of life. And that's what's kind of helped drive this interest in biosignatures and how they get preserved and where they get preserved in these biofilms that then are transitioned into our center. Is that hopefully we might be able to use different tools other than just visually looking for a microfossil. Maybe we need to look for concentrations of elements that would help to reinforce and better inform our decisions on whether something is or is not a biofilm. And I think that's also very applicable to looking for life on Mars. Any evidence for life on Mars is going to have to be a complete slam dunk. And so we need as many tools as possible to throw at any sample that we might find. So that's also something that I think has been kind of driving my interest in trying to find new tools to use in addition to the other ones. So yeah, and in relation to what our samples and the apex chert, so they're geographically quite different. And the Dresser Formation is about 200 million years older, I think, than the apex chert. So they're not quite the same time. Of course, with caveats of error bars and all that. So there were different times and regionally different areas as well. And the apex chert stuff was definitely submarine. There's pillow basalts everywhere and this huge chert veins that were coming up, and you would have to have a submarine setting to get those sorts of things buried in pillow basalts.
Yeah.
So hopefully that helps answer your
Yeah,
question.
no, absolutely. Thank you. Yeah. I'm glad to hear you say that too, because I was just talking to Greg Rotalik and he was trying to convince me the apex chert was subaerial. So
Having walked on it, I would I would disagree.
Yeah. Well, we can talk about that later too.
Yeah, absolutely. Yeah. And of course, so full disclosure, Andy Shaya was a student of Bill Shop's. So, we can still be friends. Just not Bill, because he'll hate you if you disagree with him.
Oh, great. Okay. So now I think that is the last hand up. Anyone else want to chime in just by unmuting? Hearing none. Okay. One more. One more final round of applause.